and we'll jump into it. Okay, so this is kind of where we left off. We didn't get too far into anything, but we were talking about how life is broken down into two main groups, eukaryotes and prokaryotes. And we said that prokaryotes don't have a nucleus and they don't have membrane-bound organelles. They're very simple. So in this prokaryote here, it does have ribosomes, which are used to translate the RNA into protein. But the DNA itself are just free floating inside that cytoplasm within the cell. It's not packaged up into a nucleus. It doesn't have organelles like the eukaryotes in this middle panel. The mitochondria, the ER, the Golgi, the nucleus, okay? And so that's what distinguishes bacteria from things like fungi and worms and humans, okay? We are all eukaryotes. The bacteria are prokaryotes. And then we talked about viruses being neither eukaryotic or prokaryotic because they're particles. They're not considered to be alive. And as a result, you have just a nucleic acid, whether it's DNA or RNA, depends on the type of virus, that's packaged inside this protein shell, which is called a capsid. And then some viruses have envelopes on the outside, which is actually made up of the host cell membrane. So it's a combination of lipids and sugars that that virus uses when it buds out of the cell. Okay, and this novel coronavirus that we are dealing with is an enveloped virus. So luckily, we'll get into this later, but enveloped viruses tend to have a much shorter survivability outside of a host because these envelopes don't last long outside of a warm body, right? Because again, it's made up of the cell membrane, and cell membranes used to being inside a warm host. So these viruses that are enveloped tend to break down shortly after being outside. They don't live on surfaces very long, hours to mere days. Whereas unenveloped viruses that are just this protein and nucleic acid can survive for a long time. A lot of them are found in the soil, and that protein shell is very protective. Okay. We talked really briefly about the 6 main types of microbial diversity. And one is bacteria, obviously. That is one we're all familiar with. This is a Mycobacterium tuberculosis, which is the agent that causes tuberculosis. It's a rod-shaped cell, so it's a bacillus, which we'll talk about when we get into morphology later on. But again, it's like this rod. It's an elongated shell, shape, whereas circular bacteria are called cocci. Okay, so they can be round, they can be rods, they can even be like spirals, which are called spirochete. They come in different shapes. And that's how we categorize these bacteria. We categorize them by their shape and also by their staining patterns, which we'll get into later. If they stain gram-positive or gram-negative, that's going to indicate what type of bacteria it is. Fungi. This is an image of Histoplasma capsulatum, which is one of the fungi that causes histoplasmosis. It's a eukaryotic organism, and these fungi are non-obligate, meaning they don't have to have a host to survive. They are opportunistic pathogens. So what that means is they're fine living on their own. But if they encounter a host that has a compromised immune system because it's either being obliterated with radiation because the patient has cancer, or it's immunocompromised because it has some other co-infection, then these fungi will take hold, and the immune system, which is already taxed, a lot of AIDS patients succumb to fungal infections because their body, a normal host that has a healthy, robust immune system, could prevent that fungi from getting out of hand, whereas the AIDS patient cannot. Okay, so it makes fungi kind of the jerks of the microbial world because they don't need to infect a host, right? But they do it if they can. Algae. So algae is another type of microbe, and this is a Spirogyra image, but there are all kinds of different algae. Diatoms are really cool algae. I don't know if you've ever seen any pictures of diatoms, but they look very geometric in their shape, and they are translucent but have different colors and refract light differently. It's really neat. Algae are going to be photosynthetic, so like plants, but they are not plants. Viruses, right? This is herpes. Viruses are another type of microbe, clearly, right? But unlike the others, they are not considered alive. And we'll talk about viruses in a couple weeks. Protozoa. So again, I mentioned these. I said that the protozoa are commonly the agents that cause malaria. So falciparum is a common parasite that infects humans to cause malaria, transmitted from mosquitoes. And then Toxoplasma gondii is another common one as well, or well, common for protozoa. Helminths, these are the worms. We don't have a whole lot of issue with microbial worms in developed countries, but in developing nations, these helminth worms are quite burdensome. In fact, they fall into the category of neglected tropical diseases because these are diseases that often occur in the tropics, and they're neglected because there's very little funding to research treatments because these things don't impact many of the areas of the world that have these big research institutions that are looking into diseases. So they're called neglected because they're vastly overlooked in the research community. And so here's just a summary of all these different types of microbes. The apicomplexan, oh, that's this last one here. That is, technically that's a protozoan also. They've sometimes in classifications, they will break the apicomplexan, which is the malarial parasite, outside of the protozoans. Just so you know. So if you find, sometimes there'll be 7 categories instead of 6. Okay, when we talk about how big microbes are, we have this representation here, this figure, and none of these images are to scale in the picture. So if you look here at the top, there's a flea. These cocci bacteria are much, much smaller relative to that flea, but of course we got to be able to see everything. So we look at this top picture, and as you see, this first green line here, that's what we can see with the naked eye. So you can see a lot of roundworms, and you can see some fungal reproductive structures like this sporangium, which makes the spores that then are released. But when you start going below that threshold, we get to things that can't be visualized with the naked eye alone. And that's these protozoans, the algae, the actual individual spores for mold. So if you can see all the spores together in this sporangium, that's one thing, but an individual spore is so small that it's going to be requiring a microscope. A spirochete, that is a shape of a bacterium, as I mentioned. So spirochetes, rods, and cocci range anywhere from 2 microns to 10 microns, okay? Does anybody know, like, how big a cell is, like one of your human cells, what the range is for that? No? Any guesses? Okay. No one's brave enough. Well, a cell is usually anywhere from 100 to, or 1 to 100 microns. So anywhere from down here to up at just the threshold of visibility. Now, typically, we're going to talk about 30 to 50 microns for a cell. Okay, so in this range. Okay, so so these microbes are about the size of our cells, which makes sense because, again, bacteria is a single-celled organism. Once we get below that, though, then we get to these submicroscopical thresholds where you can't just put it on a light microscope and see it. You need a fancy electron microscope. That's when we get into viruses, okay? Viruses you cannot see with a microscope. You can't see individual particles. Now, if you have an infection, if you have a tissue that's been infected with virus and you want to stain the proteins that virus makes with a fluorescent marker, then you can kind of visualize it in the cell, but you can't individually visualize one single particle. You need an electron microscope for that. And this gives you kind of a range. So, you know, basically these viruses can be anywhere from about 200 nanometers. So, so pox virus, herpes virus, those are the big ones. But then when we get down to polio virus, that is 10 nanometers. So that's going to be much smaller. And to give you reference, you know, proteins are usually about 1 nanometer. So just a little bit bigger than your typical sized protein. Right? And proteins are made in your cells and they carry out all kinds of different functions, but that's kind of the scale of things, all right, when we're talking about these microbes, where they fit, right? So the helminths and the fungi tend to be some of the bigger microbes, followed by then the protozoan and the algae, then bacteria, and then viruses, okay? And I've added a couple slides here that just give you a little bit higher resolution of what these are. If you want to look at this, this is a metric chart. Do you have to memorize this? No. I'm not going to hold you guys accountable to this information for a test. Like, you know, I'm not going to make you know that a mold spore is around 10 to 20 microns. But I want you guys just to appreciate these sizes, and so it gives you an idea as we learn about these microbes where they fit on that spectrum of size. Okay, this goes down here. Any questions on that? Okay. So you guys have probably had other biology classes and you've talked about nutrient cycling and energy cycling, and some of that, a large part of that, is facilitated by microbes, right? Whether or not these microbes are breaking down dead organic matter and releasing nitrogen into the nitrogen cycle, or carbon for the carbon cycle, breaking down CO2 into oxygen and then carbon that can be used for biomolecules. Microbes play a role, a big role. Some of them are photosynthetic. Okay, so they're going to undergo the conversion of carbon dioxide into organic material, into sugars. Others are decomposers, thankfully, right? If we didn't have microbes breaking down everything that died, it would look catastrophic on the planet. We'd have all this rotting, dead, animal corpse. So and detritus from plant biomass, right? So microbes are essential in not only breaking things down, but then breaking them down into nutrients that can be utilized by other life forms that are non-microbial, right? So they're important. Not all microbes are bad. And again, the majority of them aren't bad. Very few microbes are actually bad for us or for our animals or our food. The vast majority are helpful in some way or another. Here's algae, right? Not good if you have your own pond and you want to keep it nice and pristine and look beautiful, but also good in many other ways, because algae is being used as an alternative source of fuel. There are these biotech companies that are growing these large vats of algae and using them to produce oil that can then be used as a source of energy rather than fossil fuels. Here is a fungus, and I believe this is a little tomato that it's on, but fungi, the ones that aren't opportunistic pathogens, are decomposers for the most part. Okay, they break down things into nutrients. They break down these rotten tomatoes into things that can be used by other organisms, and themselves included. Right, so if you have a compost pile or a bin, you will see it break down more readily in the summer than in the winter, right? Because if it's not warm enough, the winter's going to kill off a lot of these microbes and little nematode worms and stuff that are breaking this stuff down. Here are cyanobacteria, the red cyanobacteria that are found in the Arctic. So as we said, microbes are ubiquitous, and they don't just live in, the nice places on Earth. They can be found everywhere. They're found way deep down in the Earth, in these places that were once considered to be impenetrable by life. They can live in hydrothermal vents where they feed off of the sulfur that comes out, that converts the sulfur. They can live here in the Arctic where, temperatures are well below freezing, that would not be considered conducive to life. Yet these bacteria have adapted a way to exist. All right, question for you guys. Let's see. Which of the following does not describe a fungus? Does not. It contains a nucleus. It has ribosomes. It's useful in decomposition, or is photosynthetic.
D.
What?
D, not photosynthetic, or it's not photosynthetic.
D as in dog. It is not photosynthetic. That is correct. Remember, fungi are not plants. Some have a very close relationship with plants. The mycorrhiza that form on the roots of plants will have this symbiotic relationship where the fungus provides the roots with nutrients from the soil, and in turn the roots supply the fungi with needed sugars. So it's a your scratch my back, I scratch your back situation between these two plants, these two species of plants and fungi. Yes, it has a nucleus. They're eukaryotic, remember? So they have a nucleus, they have ribosomes, and in fact, bacteria have ribosomes too. They are useful in decomposition, but they aren't photosynthetic. We talked a little bit on Wednesday about this, the benefits of microorganisms to humans, whether it's in biotech, to make foods, drugs, and vaccines, or genetic engineering, manipulating the genes of organisms to make new products. The example I used was insulin, inserting the insulin gene into a plasmid and then cranking out insulin that way. It's also used, they've used genetic engineering to make new bacterial species, where they've essentially just spliced together a bunch of genes of other different bacterial species to create a new distinct form of synthetic bacteria. As a, kind of a Frankenstein proof of principle that we can do these things. Your GMOs, right? All your genetically modified organisms in your food that you may or may not consume are genetically engineered using microbes to splice those genes in where they need to go. And then bioremediation. So there are bacterial species that break down oil. So when we have oil spills, oftentimes they will spray out this aerosol of microbes that then land on the oil and start breaking it down little by little. So it can be used for cleanups. I think, researchers are trying to experiment with ways to find bacteria that can withstand radiation and then can break down those radioactive materials to hopefully speed up that rate of decay, because if you haven't had chemistry, radiation of some radioactive elements takes a very, very long time. Uranium that we use in power plants takes a very, very, very long time to break down. And so if we could speed that up somehow, that would be great, rather than just burying it in the ground. Lifestyles of microorganisms. So again, most of them are free living, harmless to us, mostly beneficial. And that said, some have close relationships with other organisms, whether it's parasitic or mutualistic. Parasites are going to live on the outside or the inside of the body, and it's going to cause damage to the host in some form. It may be lethal, it may be non-lethal, but there'll be some kind of, usually there's some kind of deficit that's caused by a parasite, okay? That's what makes it, by definition, parasitic. A lot of the microbes in our gut, like this paper that we'll talk about on Monday, are not parasitic, they're beneficial, they're mutualistic, they provide us a service. The bacterium in the paper breaks down lactic acid, but a lot of other ones break down fibers and things that we can't digest on our own because we don't have enzymes to do so. The bacteria break down those materials and so that we can absorb the nutrients from them. Pathogens, a little bit of terminology for you guys, which you will need to know what these things are. A pathogen, well, that's just a microbe that does harm, okay? Pathos is, basically disease, and gen is to generate, so it generates disease in the host. We know of about 2,000 different microbes that are harmful to humans. And these different microbes cause up to about 10 billion new infections per year worldwide, okay? Which is interesting because there aren't 10 billion people on the planet. So that means people are getting infected and by more than one bug a year, right? Which makes sense because Especially if you have kids, you realize how sick they are, and then how sick you become as a result of them being sick. And then you send them off to their daycares, which are little petri dishes in and of themselves, and then they bring that back to you. 13 million deaths from infections worldwide. So the vast majority of these new infections aren't lethal, right? 10 billion, yet 13 million die. But still 13 million isn't a small number, right? And it's gotten way better in the last 100 years now that we have things like sanitation and vaccination and antibiotics and just better hygiene. This number used to be a lot higher before the 1900s. An emerging disease. So that's COVID-19, right? SARS coronavirus 2 is an emerging disease, a newly identified infection. Zoonoses, that is also SARS-CoV-2. It's an infectious disease transmitted to humans by animals, okay? We'll talk about COVID later on in a couple weeks, but as far as they can tell, it seems to have originated from a bat and maybe a pangolin, okay? But regardless, it came from an animal, okay? That's zoonotic or zoonoses. Re-emerging diseases are older infections with increasing recurrence. So think of things like measles nowadays that are that were once, well controlled because of vaccination, and then in the last 20, 30 years, when certain people have chosen not to vaccinate, we see surges in these things. So they're re-emerging. There are also things like the plague, right? I think there was a case of the plague by here in Tahoe, a hiker thinks the story I believe is that the hiker was with their dog, and maybe the dog got bit and by a flea that had it, and so the plague was once a very, very deadly disease, but now still tends to come around every so often. Okay, and that would be a re-emerging disease. Emerging is new, re-emerging is coming back. Any questions on this so far? Make sense?
I have a quick question.
Yeah.
Would plague really be re-emerging because it's not increasing reoccurrence? I mean, like, we're not having an explosion of it. Sometimes there's isolated cases. Is that called something else, or is it still considered re-emerging versus like measles? Re-emerging
is a pretty umbrella term. So okay. It doesn't have to be an epidemic or a pandemic to be considered re-emerging. It just means a disease that went into quiescence for a while and then comes back. So not continually circulating in the population.
Gotcha.
So things like influenza and other coronaviruses that aren't deadly, that cause the common flu or the common cold, those are not re-emerging because they're kind of just always in circulation. But these microbes that come in and then kind of go away for a little while and then come back doesn't have to be widespread. It's just their circulation patterns. Good question, Christine. Okay. This is a summary of the major offenders of new cases and then deaths per year, and you don't need to memorize this. If most of the tables in my lectures, you won't have to memorize them unless I specifically say. I feel like these tables are good to give you a way to kind of make sense of what are the major players and not, but I don't expect you to know that. Yeah, it might be good to know that there's a lot more cases of the flu than tuberculosis, but do you need to know that there are 3 to 5 million cases per year? No. Versus, 9 million for tuberculosis? No. Okay. But this is it. So you can see a lot of these are viruses, right? So influenza, HIV, measles, dengue fever, hepatitis. A lot of them are viruses. Malaria, that's going to be the apicoplexin. Then you have tuberculosis and typhoid bacterial. But this is what we're dealing with typically year to year. These are the worst microbes for human disease. Okay. And then interestingly, so if you look at the top causes of death in the US and worldwide, blue is going to be communicable infectious disease. Yellow, chronic disease. So not necessarily something that is caused by a microbe. It could be heart conditions, cancer, diabetes, etc. And then six, I don't know where that came from. Green, color green, is injuries. Okay, so trauma. And so if you look from left to right, low income countries, you have the majority of deaths are caused by infectious disease. Whereas if you go all the way to the right, high income countries and upper middle income countries, very, very low, right? And we know the reason that's because of access to medical treatment, better sanitation of water and living conditions make those diseases go way down. Now, interestingly, if you look at the chronic disease, it's higher in the high income countries than the upper middle. Anybody want to guess why that's the case? This is not micro related, but just I think an interesting data point.
Excess in lifestyle,
Yeah, like what?
food.
Food, like what kind of food?
High fat.
But yeah, right? So you're going to have a lot more chronic disease in high income countries relative to upper middle income because of the access to the lifestyle that leads to those chronic conditions. Presumably, like you said, high fat diets, high salt, lots of maybe excess alcohol. So it's a lifestyle thing, which I think is interesting. Now, the chronic diseases are also very high in these lower middle and low income countries, but for different reasons. The chronic illnesses aren't necessarily from a caused by a dietary perspective in terms of eating too much. It could be the opposite. It could be inadequate sources of nutrition that are driving these chronic disease. So it's, same issue, but the opposite reason causing that. Here are the neglected tropical diseases that I mentioned. As you can see, they are mostly worms. Okay, so ascariasis, hookworm, river blindness, where if you see my cursor, there's and I apologize. I know that this lecture runs during like the lunch hour. Hopefully you may not be eating right at this moment because this isn't particularly appetite inducing, but here's an eyeball that has a worm that has entered into it and is now wreaking havoc in that eyeball. And it's called river blindness because these worms are acquired through the river. People go to the rivers to bathe and swim, and then they get these worms that enter in their eye. Lymphatic filariasis, you may also know it as elephantiasis. These are swelling of the lymph nodes as a result of helminths. So, with the exception of malaria, most of these are all worm induced, and malaria is not really a neglected tropical disease. because even though it is highly prevalent in the tropics, it also happens elsewhere. So we have much more funding involved in treating malaria than we do any of these other ones. Okay. Let's take a 10-minute break, and then we'll get into the history of micro. Okay, so what time? It's like 12:51. Let's start back up at 1 o'clock, and then we'll have 25 minutes to finish. Hopefully, we'll finish this slide deck. I'll stick around if you want to use this time to ask any questions about what we've covered so far today or on Wednesday, or any other questions. Just go ahead and shoot. Otherwise, take this opportunity to stretch your legs, bathroom break, rehydrate. Okay? All right.
I do have a quick question.
Yes.
It's Britannia. So I think you mentioned we had a quiz on Monday, or was that next Friday or Monday?
Next Friday.
Is there a chapter we have to read, or is it just going to be based on your lectures?
Based on the lectures. Now, if you have the book, you'll notice that the lectures align with the book very well. So that if you want to read the book to get a better understanding, or a more broad understanding, that's fine. But there won't be any questions on the quiz that I don't cover in the lecture.
Okay, thank you.
And the way it'll work is it will be the quiz will be at 12 o'clock. And you'll have, I used to, when it was face-to-face, I'd give students 10 minutes. So I think 10 minutes should be enough. These are short quizzes. It'll be timed, so the quiz will be open between 12 and 12:10, or maybe 12:15. I'll get your feedback and see, if that seemed like a good time. But and it's just up to you guys to make sure not to cheat, right? So I feel like if I give you that 10-minute window, it's not enough time to look up all the answers in your notes or to the slide deck. So that's my way of and that's how I did it before when it was face-to-face anyway. But that's how these quizzes will work this quarter. And same with the exam. The midterm and the final, you'll have a period of time to take it. And then, it's up to you to put in the effort to know the material and do it with honesty. Okay. Any other questions?
Hey, Doc, unrelated, I'm having some technological problems here. I had a friend calling in and my whole screen just shrink into iPhone size.
Uh-oh.
So I'm trying to get my screen back.
Mm-hmm.
If I fall out of Zoom, can you invite me back in? Is that?
If you fall out, you should be able to just click on that link again and rejoin.
Okay, cool.
Yeah.
Thanks.
Yeah.
Wish me luck.
Okay. Hopefully, some of you are already starting to think about potential topics for your research proposal. There are different ways, if you're struggling for a topic. Chances are, if you read the news, there'll be something that comes up. So just this week, there were a couple articles microbe related. In China, there was an outbreak of brucella from this biotech company that was making vaccines against brucella. They were using expired disinfectants. And so the disinfectants weren't doing their job. And then they were blowing it out into this waste fan exhaust system. They were blowing out their waste brucella that they were using to create the vaccines, but they didn't decontaminate it first, and then it was in the air, and then all these people were breathing it in, and they got this outbreak. So that was just a headline this week on a news platform. There's another one. There's this Eastern equine encephalitis virus that it seems like every year now there's a little bit of an outbreak in the northern Midwest region, and so another outbreak just happened. Not a huge like epidemic or anything, but 4 or 5 people then becomes 10 people, and some live and some don't, and so if you look at the news, kind of keep your eye out for microbes that way. Another great way is if you're on social media to follow certain journals. So the 2 really big top tier premier journals are Nature and Science. Science is the US version, and Nature is the English version, meaning over in England. They're both in English.
Hey, Professor, did you say varicella at first?
When? Oh, varicella?
Your first topic. Yeah, did you say there was an outbreak of
Oh,
varicella?
brucella. No,
chickenpox.
not varicella.
Oh,
brucella.
Oh.
Yeah, brucella. Yeah, it's
okay.
sounds
Thank
similar.
you.
It's bacteria. Yeah, so a bacterial outbreak, which most vaccines are against viruses, but there are some vaccinations against bacterial infections. Yeah, so if you follow Nature or Science, then they're always like posting on Facebook and Instagram and Twitter certain highlights of articles, and Nature has a Nature microbiology offshoot. So there's all kinds of ways to come across topics by just doing daily habits like checking the news and checking your social media, and then that way as you're scrolling in your feed, something pops up and you can see it that way. So those are some tips that I recommend for those of you who are struggling for a topic. You can also look at the book, just start flipping through the microbiology textbook if you have it, and just see if there are certain microbes of interest, and then go from there. Maybe you've experienced a certain illness from a microbe, or a family member or a friend has had some disease that's microbe based, and then you just say, okay, I know somebody who had a close call with influenza. So if you pick influenza as your microbe of interest, that's not your topic, of course, but then you can start looking at research papers that have to do with influenza. And if there are any papers that you can't get, like I said on day one, email me them and I'll see if I can get them. Hava, I think, also mentioned that JSTOR, JSTOR, has an open access portion that is pretty comprehensive, so you can check that way. Unfortunately, not all scientific articles are open access, and some of the more rigorous and important studies are oftentimes published in journals that have very expensive subscription costs, so it makes that very challenging. But okay, any other questions? Hopefully the lab is making sense and going well.
I have a quick question. I'm not sure if this is intentional, but on the research proposal, when you try to open the template, it says I don't have access to it.
That's not intentional. Thank you. I'll look at that and see why that's the case. maybe it's not, I mean, that page is published, so I don't know why that's not.
Yeah, it says you don't have access to view this resource. We have let your instructor know to review this link as soon as possible.
That is a dirty lie. No one told me anything about that. Nah, it's probably some strange little loophole thing that I just didn't click right. See if I can get there real quickly and look at that. And if not, I'll correct it. Yeah. I'll have to look into that. Thanks for bringing that to my attention, though, Sarah. I appreciate that.
Yeah, it's actually doing it for both of them too. I just tried clicking on the link that says a good peer review requires disciplinary expertise. It's not letting me open that one either. So I don't know.
Okay. I will. I'll probably just delete those and then repost them and see if that does the trick. Okay. After lecture. Okay, let's hop into it again. How many more slides do we have? Okay, I think it's conceivable. I think we can do it. Next week, I believe we're talking about the immune system. Typically, this isn't the order that I have my class, but what because we're talking about COVID-19 in a couple weeks, I wanted to give you guys the information on host microbe interactions and how that works so that we can have a better understanding of that topic when we get into COVID-19. So next week we'll be talking about the immune system and host microbe interactions, how the hosts battle the microbes and the microbes correspond. And it's really just like a warfare between the two, ongoing all the time. One comes up with a better defense and then the other finds a way to break that defense or come up with its own defense or its own attack that's even better. Okay, so let's get started. And let's get me back to my lecture slide deck. Okay. I mentioned earlier that the golden age of microbiology is basically 1850 to 1950, and that now we're kind of in the second golden age. But it really dates back to before that as well. It's, for the last 300 years, microbiologists have been making contributions to this field that have enlightened us in not just microbes, but life and how life works and the origins. It all started with microscopy, right? We didn't know about microbes until we had a microscope that could allow us to visualize those microbes, which is why viruses were some of the last ones to really be definitively discovered, because they were smaller than these rudimentary early microscopes could visualize. The scientific method allowed us to rigorously test our hypotheses and not just say, well, I think that person is sick because they are possessed by a demon, right? Or they practice witchcraft, or, these things that could not be experimentally validated. And as a result of microscopes and the scientific method and understanding microbes, this led to the development of medical treatments that improved patients' outcomes, that prevented patients from getting disease to begin with, right? And then all of the techniques in microbiology that we've talked about, whether it's making sure that our food's not contaminated or exploiting microbes to produce things for us. All of this stuff has piggybacked on literally hundreds of years of endeavor. And so you know, early on, you're probably familiar, you probably learned about this at some point, but there was this belief in spontaneous generation, right? That life could just spring forth from these vital forces, okay? And a lot of times it was thought that it came from non-living or decomposing matter. Like flies came from manure, right? Because you see flies flying around your horse dung, and then all of a sudden there are larvae in that. So it must be that that's where it came from, right? Louis Pasteur, who came up with the process of pasteurization, was the one who really disproved this concept of spontaneous generation. And you guys have probably heard the story where he put cheesecloth over this meat so that there couldn't be anything getting into the meat. And then another vat of meat, he just left it open. And of course, flies came from that was uncovered. But the dish that had the cheesecloth, no flies. And so that was kind of the seminal breaking point that you just can't get something from nothing, right? And when you kick some widely held belief out of existence, you must replace it with something, right? It's far better to say, I don't think that that is the case, because I think that this, what I'm going to tell you, is the case, right? It's much more compelling than if you just are a naysayer. And so he not only disproved spontaneous generation, but then provided the theory of biogenesis, that living things only arise from other living things, okay? And that seems so obvious to us now. But in the 1800s, that was something that people, had to wrap their mind around. And this is after Charles Darwin, published his Origin of Species. So like even at that time, when the world was gripping with this concept of evolution, whether or not they bought into that, some people still thought that you could have things just come from nothing. All right. So we live in a very different time. I mean, a lot of these things are still probably disputed, right? But we know so much more now than we did just a couple hundred years ago. And it's baby steps, right? It takes a long time to prove something of this magnitude, and then even longer for that to be internalized into the minds of society. Then Anton von Leeuwenhoek, he's the guy who came up with the microscope. So that labster that you guys did on Wednesday, or hopefully you're going to do soon, and I know that there were some issues with it crashing. There were some issues with maybe some of the questions that were ambiguous. I want you to know that, especially for these first few labsters, if you do it and you tanked it and your score is bad, I will update it for you. Okay. I don't want anybody to have a bad score on their first few labsters because they're trying to figure it out and navigate it. Okay. So I can clearly look in gradebook and see if anyone's done poorly. If you have, or if you feel like you got, the short end of the stick for some reason, shoot me an email. We'll discuss it. We'll work it out. Okay. I don't want you to just go through it and just click idly and not think about, the questions and say, oh, well, I got 5 out of 10. But I also don't want you to have a bad score on Labster. That's not the point. Anyway, Leeuwenhoek, he's the one who came up with the first microscope. And it was just one single lens, but that single lens could magnify things up to 300 times, okay? And this is basically what it looked like. It had this holding station here that contained the lens, and then there was a little needle that you would put your specimen on. And then you could move that needle, if this is the lens here, you could move that needle closer or further away from the lens by turning a little screw, and then you just hold it up to your eye. So very rudimentary, very basic, but it opened up a world that no one had ever knew existed up until that point, right? Something as simple as this, as a piece of glass and a screw, shed light on this microbial, underrealm that no one knew existed. And here's actually, this is an illustration from Leeuwenhoek that he drew, and this is what he saw, right? He saw these little tiny things, and I believe Leeuwenhoek was the one who coined the term cell. I could be wrong, because I think he was a monk, and he lived in their quarters were called cells, and they were small. And he looked under there and he saw these little things, and they looked like the little monastery cells. And so that's where we got the name cell. Don't quote me on that, and that won't be on the quiz because I'm not absolutely sure, but I think that's how the story goes. Okay. Scientific method. Now, again, we've all learned this, but I think it's so extraordinarily important to really understand it, not just learn it, but understand it, because this is what makes the world go round. This is what informs us of how the world goes around. And this lack of understanding of the scientific method is why there's so much pervasive scientific denialism in the world right now. It's why so many people, I think, deny the tenets of science that are pretty much a consensus, right? And, we won't go in a debate in this class. Hopefully we can all agree, but, the benefits of vaccination, the reality of global climate change, and the human involvement in that change. Even GMO use, right? Some people are completely anti-GMO, but it's because of GMOs that certain people in Asia can get their beta carotene, if you've heard of golden rice, right? That they can get the nutrients that would otherwise not be present in just regular rice that could lead to blindness. There are people trying to make genetically modified, oh shoot, what are they called? It's a starchy, it's like a potato in the sub-Saharan African desert. Oh, it's on the tip of my tongue. Anybody know what I'm talking about? What am I thinking? Nobody know? Okay. Taro. Taro? No, maybe. Yeah, maybe taro. Anyway, not important. But
Taro's Japan.
What is that?
Taro's in the Pacific.
Okay, not taro. It's basically like, it's just a starchy root. Okay, it's like a potato. It doesn't have very many nutrients, but it grows in an arid environment where they can't grow anything else. And so they're trying to add in different genetically modified genes to add in nutrients so that people in sub-Saharan Africa can, get nutrients and not be malnourished. But, there's this thought that genetically modified organisms are all terrible. Not all of them are good, right? But not all of them are bad. We might talk a little bit about the idea of genetically modified mosquitoes, which they've already generated and they are releasing in certain parts of the world. And I think Florida did this, where it's a genetically modified mosquito that can't reach maturity, essentially. And it's the mature mosquitoes that mature females that cassava, there it is. Thank you, Natalie. Cassava is the starchy plant. These mosquitoes can't transmit malaria because they don't reach sexual maturity. Okay, so anyway, all of the rejection of very well-established science, I believe, is a symptom of not really appreciating and understanding what goes into the scientific method, okay, and how robust something has to be to make it through each step. Okay. You have to, you have to publish your results so they can be repeated, because if you can't repeat it, then is it really is it really true? Okay. And I hinted at this a little bit when I talked about the woman who was interviewed in Plandemic, and how she thought that she had found the link to chronic fatigue syndrome, and it was caused by a virus. And then she published, and it got global attention. And then the researchers like, oh, great, let's test this. And no one could repeat it, right? And then that's how they found out that it was not reproducible, because it was not accurate. It was contamination, right? So you publish it, they repeat it. If it's compelling enough, it becomes a law or a principle, right? Okay, so if something is a law, that means it is very well regarded, it's very well reproduced, and we have a high confidence that it is actually true, okay? But if it goes beyond that, if it goes to the level of theory, that means it's supported by multiple lines of evidence, has undergone highly rigorous scrutiny, and then it transforms or transcends from a law to a theory, okay? I can't tell you how annoyed I get, and it's kind of silly, but when people say, whoa, it's just a theory, that just wrinkles me, you know, to the point of wanting to turn off the TV when I watch these murder mysteries, and you know the detective says, well, I have a theory that it's this. It's like, oh, well, that's just your theory. And you know, semantics, right? But a theory is not just what you think. Yeah, if you're a detective, you have a hypothesis. You think that, you know, Mr. Mustard did it in the library with a candlestick. It's not a theory, right? And they use these words interchangeably. So when we say that there's a theory of evolution, when I see these arguments to it that, oh, well, evolution is just a theory, that shows how little they understand science to begin with. Because if you're saying it's just a theory, you're saying, oh, well, evolution is just a hypothesis that's supported by multiple different scientists in multiple different fields and has undergone highly rigorous scrutiny over and over and over again and has been reproduced. And so, yeah, it is just a theory. Fine. Okay. So don't make the mistake in my class of using theory incorrectly. Okay. And I will probably test you on that. There'll probably be a quiz question or a test question to see if you know what a theory is and just how powerful a theory is, okay? All right, I'm gonna get off my soapbox with that and move on to—
I have a quick question.
Yes, Christine, shoot.
I'm sorry. You know, this seems like a simple concept, but for some reason I get tripped up with deductive and inductive.
Okay, well, what do you think?
Well, you have the if-then on the deductive, but I honestly, I don't know why. Like, I can get concepts more complex than these probably, but for some reason I have trouble with those two.
Yeah, so inductive is to induce, right? So if you're inducing something, and there might be some evidence, but it's really grounded in more of a hypothetical form. Okay, so inductive reasoning means I'm seeing these connections, okay, and I'm just going to put those connections together because it makes sense. So it's somewhat supported by evidence, the evidence that you make with your observations, but it hasn't been, but it's you're the conduit making those connections. Whereas deductive is taking two things of evidence, putting them together, and then seeing what happens as a result of that. And by looking at that, you can then deduce. You're not inducing, you're not kind of driving it. You are a passive observer and saying, okay, well, based on all of these things coming together, the clear observation is this. Does that make sense?
Okay, so deductive, you're more the passive observer of what happens. And then with inductive, you're not passive, you're an active predictor.
Yeah, I mean, so to a certain extent, I mean, deductive is not completely passive because you're playing a role. But my understanding is that when you have deductive reasoning, so like that was Sherlock Holmes' big shtick, right? He was great at deductive reasoning. So he'd look at all these multiple lines of evidence, these clues, and he'd weave it together, and whatever the tapestry showed, that must be what the truth was. Okay. Whereas inductive is saying, okay, I'm going to take all these threads and I think that the tapestry is going to look like this, but I haven't woven them together enough to have the tapestry in front of me. Does that make sense?
Okay. Yeah, move on, and I'll look into it more on my own. I don't know why, but it's weird.
I
Okay.
can say that a little bit differently if you'd like.
Yeah, let's hear it.
Okay.
So in a deductive argument, if you have certain premises, then once you put them together, there's no possibility except for the conclusion. So there's a logical force that when you have certain premises, they end up at the conclusion, and it's not possible for you to not have the conclusion. So like 2 + 2 = 4. That's like, I mean, that's just an a priori thing. But like if you have 2 and then you add 2 more things, it's not possible for you to have anything other than 4 things. I mean, I guess if you like, you can mix things together, but that's kind of the point. With an inductive argument, you're making inferences. It's more like conjecture. You're taking evidence and thinking about what the most likely thing is, and like doing the best with the information you have. But with deductive reasoning, a sound deductive argument can't end anywhere except for the conclusion. It's like completely ironclad. If that makes sense.
So one's more like a hypothesis, the other is like law.
That's a pretty good way of putting it.
Yeah. Thanks, Eric. Yeah, that was great. I appreciate that. And I encourage you guys to jump in at any point. We all learn together, right? And this is a collaborative process. So yeah. So hopefully with some of those, with those 3 descriptions that becomes a little more clear. The scientific method is essentially, as you guys know, an observation, hypothesis, experimentation, results. You know, you do your analysis, you form conclusions, you publish. It gets reproduced to the point where it's a law, or it gets reproduced and validated and verified independently until it becomes a theory, or it becomes rejected when we have other information that tells us a clearer picture, right? So, I mean, that's the simple form, but really it doesn't work that way in practice, right? You have a hypothesis, you design an experiment, and it fails, right? And so you have to optimize it and troubleshoot. And then it gives you a different outcome than what you predicted. And so, you know, this says results are not consistent with predictions, so you have to form another hypothesis, right? Or you have to alter it a bit. And so it's this circular thing that goes round and round until something shoots off that gives light on an understanding to things that we didn't know previously. And that's why it's so powerful. It's not just scientists playing around with beakers in the lab wearing coats and screaming eureka and being nerdy, right? That's all part of it. But well, in fact, I've never been in a lab where a scientist, I worked in a research lab for 10 years. I never heard anybody shout eureka, but you know, it's a very, very detailed, involved, years-long process to get your answers. And so that's the power of the scientific method. Oh my gosh, we're out of time. Oh, holy moly. Okay, well, concept. A scientific theory has little or no evidence to support it and could be best described as a best guess. True or false? Yeah.
False.
False. False. So when you watch a murder mystery and the detective shuts down some other detective by saying, well, that's just a theory, you'll know, right? And these are, I usually watch these like BBC murder mysteries or like PBS where it's in Britain where they came up with the English language and they're misusing it. So go figure. Let's see, we have a little bit more to cover, and you know, it's not much. We'll get to it. We'll rock it out on Monday morning. Hopefully I will not get distracted and go off on my tangential things, and we'll just stick to topic as best possible, and then jump into the host microbe interaction chapter. So if you do have the book, if you want to read up on that, then that's going to be chapters 13, 14, and 15. So we're jumping ahead. Chapters 1 and 3 are the chapters for this slide deck, and that'll be the stuff that's probably going to be on the quiz on Friday. So just chapter 1 and 3 material on Friday's quiz. But then we'll also be talking about the immune system, okay, next week. So thank you guys for signing in. I appreciate it. I will look into those proposal templates and stuff that didn't work, didn't give you access. And I think I got some messages from a few of you, so I'll look at those, and have a great weekend. Hopefully your experiment went well today, the lab. And if you're still needing to pick up the kit, please do so today so that we can, ideally next Friday, spend just a brief time on Zoom looking at our plates and showing each other, and get that going and keep on track. So I can stay on for a few minutes if you guys have any other lasting questions. Otherwise, have a great, safe weekend, and I'll see you Monday at 10 for our next lecture. And remember, Journal Club, okay, for lab after that.
So actually, you just mentioned the lab and how we should start it today.
Mm-hmm.
I'm not able to pick up the kit, so and I don't think I can find just Petri dishes in my local Walmart. Can I just do clear plates that I have at home?
Yes. A couple of you have asked that, and absolutely. You can even use a dish. You know, like if you have a little bowl, it doesn't have to be clear. You can use a little bowl. You don't want it super big. I mean, there's nothing wrong with it being big. It's just you have to make a lot of agar for it. But if you have a little dish, like like a ramekin or something like that, those work great. And then you can cover it with saran wrap or like cling wrap, so that it's sealed, so that you don't get any other microbes in there. That's fine. Or even like a Tupperware dish, like if you have a little Tupperware with a lid that screws on or snaps on, or a glass dish, any of that stuff is fine.
Okay, thank you.
Mm-hmm.
Professor, I have a question for how much can we pour the agar into the dish?
Yeah.
Like half of the way, or?
Well, it depends on the depth of the dish, but you really want a good, I would say, 1/2 inch. Okay. These things will dry over time, to some degree, especially in Tahoe. It's very dry up here, and so the drier the environment, the more these agar pads will dry up. So you want it thick enough that it doesn't dry up into a little thin film. Okay, so anywhere from a 1/2 inch to 3/4 inch should be fine.
Yeah, thank you. And one more question for the
Yeah.
agar. We have to make at home, or we order on.
So let's go. So you can't pick up a kit at Tahoe. You're not in the Tahoe area. Is that correct?
No, I already picked it up.
You picked up the kit.
Yeah, and I see that they have the dish and this empty in there. Is it already have the agar? I see like it looks like there's no agar inside.
Is that true?
You know, mine almost looked like it didn't have anything in it, but it did.
Okay.
I mean, when I first looked at them, it didn't look like there was anything in there, and I was worried. But when I opened it up to actually do the stuff, oh, there is something in there. So I don't know if you've actually opened it.
I didn't
because-
open it. I didn't
Okay,
open it.
try
Yeah,
it.
so
Try it
don't-
because mine, I didn't see it.
yeah, I didn't see it. I see it's very thin layer, and I thought, is that?
Could you show me? Do you have your plate nearby? Could I see?
No, I
Or
don't have it right here. Yeah.
I have mine here.
Let's see.
Pretty thin.
It's pretty thin.
Like you can barely see like 2 drops in there.
Oh.
Okay. Huh. So ideally, it would have about a half an inch of agar in there. I didn't pour these plates, but Kevin knows how. I wonder if they dried out.
So should I make it one more and then I pour it in?
Does it seem like it's like just a very, very thin, hard,
Very thin. Yeah, I
Very
see it. It's very thin layer. It look like
uh,
it doesn't have. Yeah.
that's
Mine was very thin, but it seemed like the regular consistency.
mhm.
It wasn't hard, and you could actually put a little divot in it when you did the swab
Mhm.
part from my cell phone. So the consistency seemed right. It's just super thin
Okay.
layer.
Huh. I'll talk with Kevin. He's the lab tech who made these kits. Make sure, in that case, make sure that you do put them in a plastic bag and seal the bag. You should have some plastic bags in the kit because that will at least prevent further desiccation. Because I'm hoping that these plates will go a week, right? And if they're left out, they'll dry out completely, and then you won't really see much. Okay.
And for one more, the one like before washing the hand and after washing the hand, so we can wash with the water or with the soap. And like in that we have only one column to test, like with the hand when we touch it with the soap or without the soap. And then in the table you have 2 columns. So in that case, should I do it with both or just either?
2 columns in the table.
In the table 2. In table 2, I
Left
see
index
that.
finger before, left index finger after.
Yeah. After washing with the water and after washing with the soap. So we have 2 columns. But like when we do it, we just either. So when we fill
No.
in, we do only one column.
Sorry. I believe that you draw a line down the center of the plate. And then on one side of the plate, you'll do your finger before washing. And then on the other side of the plate, you'll use your finger after washing.
Yeah.
But on the same plate.
And after washing, like with the water and without, with the soap and without the soap. So I just do only one.
Yeah. Okay. With soap or water. Okay.
Yeah.
So pick one. I would do soap because if you wash it with water, it's probably not going to be very different than not having any water on your hands. So my recommendation would be on this side, it's before washing, and then on the right side, it's washing with soap.
Yeah. So in the table, I just fill in only one.
Yeah, that's fine.
Thank you.
Yep. And hopefully, man, I'm kind of worried about this. That the plates are that the agar is so thin, but hopefully I'm going to make an announcement to have everybody put those plates in the sealed bags. It might be that only the people who couldn't get kits who live distantly make their own. That might be the most informative, unfortunately. I'm hoping the plates don't dry out, but we'll see. You know, that's the thing about experiments. Like I just said, oftentimes they fail. So this is your first learning experience of how experiments go wrong. The hard way, unfortunately. Okay.
Okay. Thank you, Professor.
But thank you for bringing that to my attention. I appreciate that.
Um,
So Doc, I did have a lab assistant after I connected with you. And in the 5th and 6th procedure there with the swabbing of the phone, the phone actually fell accidentally into the sink and got some of the grime underneath the granite there, some of that slime. And after we swabbed it with alcohol, the phone then fell into the guinea pig cage with pt2. So my results might be a little different. Just to
Wow, that's a slippery phone, Ryan.
We're a little uncoordinated in this house, maybe adventurous at the same time.
What? Just document that, because that could lead to some pretty interesting results.
Okay, cool.
Yeah.
The other thing, you keep bringing up this example of, and I'm looking for it on the internet. It's the scientist who went after a CFIDS cure, was it? Chronic fatigue cure, and she was debunked by, it wasn't repeatable. What? Do you have details on that so I can actually read the journal or article?
Yeah. Let's see. Her name.
Yeah. The name would probably be enough.
I mean, they Judy. Here, I'll just put her name in the chat. Judy Mikovits.
There it is. Thank you.
And I'm sure if you just start searching her, you could probably even there's probably a Wikipedia page that will have that paper that was retracted.
BITS, perfect. Thanks for that.
But yeah, I mean, it's very interesting. I remember, so before she was this crazy pandemic person, when I was in graduate school and we were I was in a virology lab, and I remember my mentor just anecdotally mentioning this story, and he didn't mention her name or anything, but he told us the story about how, at that point it was a learning experience for us not to over interpret our results, because contamination is so prevalent in a lab. If you have any bugs, they're ubiquitous, right? And so we try to do things to minimize that by working in hoods and having UV to kill off things. But, it just takes a little tiny droplet of something to get into the wrong tube, and then using that tube to then transfer to all your other tubes. And he used that as an example of someone who had, basically ruined their career by thinking that she had found the cause of chronic fatigue syndrome. So
one of the origins on that was actually Incline Village, 1984, and they kept it hush hush because it was spreading fast amongst a very prosperous community. They called it the yuppie flu virus at first, because there's all these type A personalities, these overachiever types, but eventually it was contaminated water. One of the water wells was contaminated, and that little bacteria, correct me if I'm wrong, had then triggered this autoimmune response with people that had genetic predispositions from people that were healthy, and a large portion of the community went bad.
No kidding.
Yeah. 84, Incline Village.
That was the big, that wasn't the big winter. Was it 83 that was the big winter in Tahoe? Big snow?
They all seemed big back then as a kid.
Probably all of them, yeah. Yeah, well, we'll talk about a little bit later on when we talk about Legionella. It's a bacterium that it got its name because of it was first discovered in a Legion hall. So it was a contaminated air conditioner. The condensation in the water got this bacterium in it, and then it blew into the Legionnaires, meeting room area, and they all got infected by it. So, yeah.
Yeah, scary story in our present time.
Yeah, absolutely.
Thank you.
Yeah, you're welcome. All right, anything else, guys? All right. Well, I'll then sign off. I'm gonna go help out my wife with the 4 kindergartners right now, and have a good weekend. We'll see you Monday.